Hello friends, dear learners and my dear student. First of all, I take an opportunity to welcome you at my T.Y.B.Sc Analytical Chemistry class. I am Dr.S.R.Labhade, working as Associate Professor in the Department of Chemistry at K.T. College, Nasik. Student, in previous lecture we discussed the factor affecting the intensity of resonance line or emission line. We also discussed the Boltzmann's equation. For this purpose, which shows the relationship between the number of or a fraction of atoms that undergo electronic transition in the flame. In addition to this, we are discussing about the instrumentation of flame emission spectroscopy, and we are discussing the nebulizer burner system, that is an atomizer. Atomizer, that is a device. Atomizer is a device that converts liquid sample into ground state gaseous atoms. And we know that atomization is the first and important step in atomic spectroscopy. Atomizer, that functioning as a atomization process. Atomization is a process in which liquid sample is converted into ground state gaseous atoms. Atomization is the first step, and that is the important step in atomic spectroscopy. In addition to this, we also discuss the thermal excitation of atom is the process of promotion of valence shell electron of ground state gaseous atom into the outer next cell of the atom by absorbing thermal energy. Therefore, after atomization, there is excitation of atom. Excitation is a process by which the valence shell electron is given to next or outer excited state cell by absorbing any energy, that is a thermal energy. We also know atomic emission. Atomic emission is the process in which the radiant energy is emitted by the atoms by the excited state atoms when those are returning to their ground state. Therefore, we also discuss the atomic emission. Another term which is related with this is called as nebulization. Nebulization is a process by which the liquid sample is converted into fine mist and transportation of it onto the burner head. Or the tip of the burner. Therefore, here nebulization is a process by which liquid sample is converted to fine mist or fine droplets or fine aerosol. This aerosol is then transported into the burner tip, and this process is called as nebulization. Nebulization is the first step in atomic spectroscopy. After nebulization, there is atomization. After atomization, there is thermal excitation, and after thermal excitation, there is atomic emission. Student, in continuation with nebulizer burner system, we discuss here different types of atomizer. Atomizers in FES or in flame emission spectroscopy are divided into two types. First type is called as total consumption burner. Or it is also called as integral atomizer. Now the construction of this integral atomizer. In an integral atomizer, fuel gas, oxidant and air, those are given through a separate compartment, and all are reached to the burner head through a separate channel. This is the construction of integral atomizer. In this case, this is for inlet for fuel gas. This is inlet for air or oxidant, and through this a sample solution is taken by a Venturi effect, and here all these three mix at the burner tip before that are burned into the flame. Therefore, it is a burner tip. In this way, the integral atomizer is constructed. In integral atomizer, the sample solution, fuel gas and oxidant are passed through separate passage to enter into the flame. In this case, the sample is taken into by a Venturi effect. Venturi effect in which along this capillary tube. This is a capillary. A high pressure gas is passing, therefore that automatically suck this sample inside the chamber. This effect is called as Venturi effect. That means here with this compressed air, sample solution is automatically sucked into the flame. There is no other provision made to take the sample. This burner utilizes total sample, therefore it is called as integral burner. It utilizes the entire sample and therefore called as integral burner or integral atomizer. In this case, the sample is taken through this capillary along with the compressed air. The high pressure air breaks this sample solution into fine mist or fine droplets, or here a aerosol. This fine aerosol is then transported to the burner head. This is the working of this type of integral burner. Here it utilizes total sample and therefore it is called as integral burner. Now we discuss the advantages and disadvantages of this total consumption burner or a integral burner or integral atomizer. First advantage is that entire sample is utilized. Entire sample is utilized, therefore have good sensitivity. Sensitivity directly related to the amount of sample atomized. Therefore, if total sample is atomized and if concentration of sample is very low, then at that lower concentration, here sensitivity can be increased if the total sample is utilized for the atomization process. Therefore, entire sample is utilized and it gives the good sensitivity. Second, no variation as total sample is utilized, no variation in results due to variation in droplet or droplet size. Third parameter, as there is no mixing of fuel gas and oxidant with the sample solution, there is no hazard of explosion. No explosion hazard in the integral burner. Now here these are the three different advantages of this integral burner. Now the disadvantages. First disadvantage, as total sample is utilized, the flame temperature decreases. Therefore, here solvent require some thermal energy, and therefore total sample, if it is utilized, then most of the thermal energy of the flame is utilized for the evaporation of solvent, and therefore temperature of the flame decreases and gives incomplete atomization. That means atomization process is not complete for total sample. Therefore, fraction of samples are atomized. Second disadvantage is that here the large size droplet clogging may possible due to larger droplets, and due to these disadvantages there is requirement of another type of burner. Therefore, second type of atomizer is laminar flow burner. It is also called as premix atomizer. Now we discuss the construction and working of this laminar flow or premix atomizer. Now we discuss the construction. The laminar flow burner is constructed as shown in figure. Here, this is the inlet for fuel gas. This is inlet for air or oxidant. Here, from this sample solution is taken. It utilizes a fraction of sample. Therefore, most of the sample is given out through the drain. These are the mixing plates or mixing battles. Here, this particular chamber is called as mixing chamber. Initially, fuel gas, oxidant as sample solution are mixed into mixing chamber. Then fine mist is formed and this mist is given into the burner head where flame is present. This is the construction of the laminar flow burner or premix burner. Name is given as a premix burner because here fuel gas, oxidant and sample initially mix in a mixing chamber and after that it is given into the flame. Therefore, very fine droplets or aerosols are given into the flame that increases the efficiency of the measurement. Therefore, in this burner, in this atomizer, a fraction of sample is utilized. The fuel gas plus air plus sample is mixed in mixing chamber and then given into the flame. In this case, sample is also taken inside the chamber by a Venturi action. Therefore, Venturi action, the sample is taken in mixing chamber. Due to longer path, most of the sample is condensed and given out from drain. Only 10% of the sample in the form of fine mist given in a flame. Therefore, 90% of sample is given out from this drain, because here, as here Venturi effect takes the sample inside the chamber that convert the sample into a fine aerosol or a droplets of different size. The larger size droplets are get condensed on these mixing baffles or a mixing plates, and only fine mist that is given in the plane, and due to this only 10% of the sample reaches to the plane. This laminar flow burner have good efficiency, and as the part of sample is utilized, sensitivity of measurement is more. But here, loss of sample through the direct train does not affect the measurement. This is because of flame emission spectroscopy is a comparative method. Whatever the flow rate for sample solution, exactly same flow rate we have to maintain for the standard solution. And then here, whatever the amount of sample given out from the tray during the aspiration of sample and during the aspiration of standard are same. And therefore, this does not affect the measurement. But here, as a part of sample is utilized, the sensitivity of this measurement, sensitivity of this laminar flow burner is less compared to integral atomizer. Now we discuss the advantages of this laminar flow burner or pre-mix burner. First advantage, due to longer path, fine mist or aerosol is given into flame. Fraction of sample utilize, therefore maintain flame temperature, and atomization process is complete. Atomization is complete, or total atomization of total sample takes place. That means whatever the sample reach into the flame, that total sample is atomized because flame maintained to a higher temperature. These are the advantages, but the disadvantages, there is a chance of explosion, in a mixing chamber. Second thing is that as a fraction of sample is utilized, decreases the sensitivity. That means if solution is very dilute, containing very trace concentration of iron or a alkali or alkaline earth metal iron, then if part of that will be lost, then very few atom reaches to the flame. At that time, sensitivity decreases. Student, this is about the two types of atomizer used in atomic spectroscopy, that is flame emission spectroscopy as well as in flame atomic absorption spectroscopy. First atomizer is called as integral atomizer, where total sample is utilized, and second atomizer is called as premix atomizer, where a fraction of sample is utilized. But here premix atomizer is superior over the superior over integral atomizer because it maintained the flame temperature and atomization process is complete for total number of ions. Student, now we discuss what are the effect or events occur when sample is given into the flame. Student, now we discuss the different regions of the analytical flame. We know that in atomic emission spectroscopy as well as in atomic absorption spectroscopy, flame is act as a sample cell. The function of the flame is number one, to atomize the sample into gaseous atoms. Second thing, once those atoms are atomized, to excite the ground state gaseous atom into excited state, and third, to hold all those excited atoms till those does not gives emission. Therefore, flame emission spectroscopy act as a sample cell. Its function is to atomize the sample, to excite the sample, and to hold the sample till it shows the emission. Therefore, the function of flame is in FES is to hold the sample for their atomization, excitation, and the emission. Therefore, it is called as a sample cell. Its function is to hold the sample for its atomization, for its excitation, and for its emission. Now here, analytical flame have these three different regions. The first region in the analytical flame, that is called as primary consumption zone. The middle one region is called as interconal region, and outer cone or the outer side is called as outer or secondary consumption zone. Now here, this primary consumption zone have a very low temperature, and color of the flame is appear as a dark blue. In this case, total sample is atomized, it is excited, as well as its emission is also occurs in this region. And this is the hottest part of the flame, having a reddish color, and it is a transparent one. Again here, outer cone have a light blue color, and temperature of the outer cone is again less than the interconal region, because this part is directly exposed to the atmosphere. The sample is given in an outer cone region, and after that it is given in an atmosphere. Therefore here, analytical flame has three different regions: primary consumption zone, second one is interconal region, and third one is the outer cone or secondary consumption region or zone. Primary consumption zone, where this is a cooler part of the flame where solvent is vaporized, leaving behind residue. The residue is further given to the intercorner region. This is a hottest part of the flame where dissociation means atomization, excitation and emission occur. Therefore, analytically, this region is very important. It is important region because FES measure the emission intensity. Therefore, the optical system is exactly placed in front of this intercorner region in the instrument. Because atomization, absorption and emission, total 3 events are occur in this intercorner region, and therefore analytically this is very important region, and this is a hottest part of the flame. Outer cone that have a little bit blue color, that is called as secondary consumption zone. This zone is directly exposed to atmosphere, so its temperature is decreases or it is low, and this is a bulk. This is the bulk region of the flame. Actually, this is a 3-dimensional figure. Flame have a 3-dimensional structure. Inside there is a blue color, that is a primary consumption zone where solvent is evaporated. Residue is given into intercorner region where dissociation, atomization, excitation and emission occurs. Therefore, analytically this region is very important. Therefore, optical system is placed exactly in front of this region, and outer cone region from which the sample, that is atomic species, is given out in the atmosphere. Therefore, this is the function of analytical flame, which act as a sample set in atomic spectroscopy. Student, with our today's discussion, you can find the answers of following questions. Explain the terms. Number 1, atomization. Number 2, atomic emission. Number 3, atomic excitation. Number 4, nebulization. Again, next part of our discussion, that is a give construction and working of. Give construction, working, advantages and disadvantages of. First one is integral burner, integral atomizer, and second one is the premix atomizer. question of flame. What is function of flame in FES? Explain the different region. Explain the different region of analytical flame. Therefore, with this today's discussion, you can find out the answers of this question. With this, we stop here. Thanks for joining me. Wish you all the best. Keep learning. Happy learning.